Welcome to today's lesson on tissue culture, where we will explore the fascinating processes and advantages of this technique in medical research and plant breeding programs. In today's lesson, we will first understand what tissue culture is. Then we will look in more detail at the specific steps involved in plant tissue culture, used to support plant breeding, and animal tissue culture, used to aid medical research. Understanding tissue culture helps explain how scientists can create multiple copies of a plant or exact clones of human cells. This insight is fundamental for innovations in developing new medicines, improving crop yields, and conserving rare plant species. Let's get started. Tissue culture is an essential technique in both plant breeding and medical research. This method allows scientists to grow small pieces of tissue from an organism in a nutrient-rich, artificial environment. Using tissue culture, entire plants can be grown from just a tiny piece of plant tissue. Let's explore how this is done for plants and animals. Here's a detailed look at the process of plant tissue culture. First is selection. A parent plant with desirable characteristics, such as vibrant flowers, unique leaf shapes, or high fruit yield, is selected. Next is extraction. Small tissue samples, known as explants, are taken from the parent plant. In the sterilisation stage, the explants are transferred to a sterile petri dish containing nutrient-rich agar. This is done under aseptic conditions to prevent contamination by microorganisms. To promote cell growth, hormones like auxins are added to the nutrient agar. The explants eventually grow into small plantlets. These plantlets are then transferred to potting trays with compost to mature into fully grown plants. Plants produced through tissue culture are clones, meaning they are genetically identical to the parent plant. This technique is highly beneficial in plant breeding programs for several reasons. One reason is cost effectiveness. Growing identical plants via tissue culture can be more economical than traditional methods. Another is high yield. Large numbers of plants can be produced from a small amount of starting tissue. The process is also space efficient, as it requires less space compared to growing plants from seeds. Plants can be produced quickly, much faster than growing from seeds. There is also year-round cultivation. Plants can be grown at any time of the year, independent of seasonal changes. Additionally, tissue culture helps prevent the spread of diseases by allowing for the mass production of plants from a single, disease-free specimen. It also plays a crucial role in the conservation of rare plant species. Animal cells and tissues can also be cultured, although the process is somewhat different from plant tissue culture. The first step is extraction. A tissue sample, such as from the pancreas, is taken from an animal. Next is separation. Enzymes are used to separate the cells within the tissue sample. Then cultivation. The separated cells are placed in a culture vessel containing a growth medium that supplies all the necessary nutrients. Lastly, the cells grow and multiply, forming tissues that can be studied or stored for future use. A key point to note is that cultured animal tissues are extremely valuable for medical research. For example, pancreatic tissue can be grown in culture to study how glucose affects pancreatic cells. This allows scientists to conduct experiments on tissues in isolation, free from the complications and external factors present in whole organisms. Detailed studies can be conducted to understand the effects of specific chemicals or environmental changes on individual tissues. In today's lesson, we examine tissue culture and its significance in medical research and plant breeding. Tissue culture involves growing cells in a controlled environment, allowing scientists to produce large quantities of identical cells, tissues, or plants from a single sample. For plants, this process includes selecting and sterilizing the tissue sample, then placing it in a nutrient-rich medium. In animals, cells are extracted, separated with enzymes, and grown in a culture vessel. Tissue culture is valuable in medical research for studying cellular processes, testing drugs, and exploring genetic modifications. In plant breeding, it helps produce disease-free plants, rapidly multiply desirable traits, and preserve rare species. Understanding tissue culture highlights its crucial role in advancing medical and agricultural sciences, setting the stage for future studies in genetic engineering, disease treatment, and sustainable agriculture.